Hello, everyone. Welcome to today's webcast, Characterizing Protein-Nucleic Acid Interactions by Light Scattering. I'm Rita Peters, Editorial Director of BioPharm International, and the moderator for today's event. Now I'd like to introduce today's speaker. Sophia Kenrick is a Senior Application Scientist at Wyatt Technology, where she supports multiple applications for Wyatt instrumentation, especially in the field of molecular recognition and biomolecular interactions. Dr. Kenrick received a bachelor's degree in chemical engineering from Arizona State University, a doctorate in chemical engineering from the University of California, Santa Barbara, and has been with Wyatt since 2010. She's also the Dean of Light Scattering University, Santa Barbara campus. So thank you for joining us, Sophia. I'm going to turn the presentation over to you.
Thanks so much for the introduction. In today's webinar, I'll be highlighting characterization tools for analyzing nucleic acids and the interactions between nucleic acids and proteins using light scattering. So we'll start off with an introduction to multi-angle light scattering, and then I'm going to present two case studies that show the interactions between DNA and their protein ligands. Throughout the course of this webinar, please keep in mind that these techniques are not just applicable to DNA, but are also to RNA, pegylated or glycosylated molecules, and engineered proteins and nucleic acids with non-natural components. So a lot of times when we think of biotherapeutics, we think of proteins or antibodies, but really all kinds of biopolymers can be used for these kinds of drugs, and especially now, DNA and RNA biotherapeutics are on the rise with the advent of things like cancer immunotherapies and cancer vaccines. So this involves delivering RNA to T cells or other immune cells and transforming them into tumor targeting agents. And the delivery is really crucial for this process. So biophysical characterization is required to understand the properties of these drugs. So for example, the delivery efficiency could be dependent on how much polymer is conjugated to your RNA. Or maybe you have a purification or formulation condition that causes your single-stranded RNA to form duplexes and other large aggregates that you don't want. What I'll show in the next few slides is that light scattering can provide key information about the size, molar mass, and conformation of these complexes. Characterizing the interactions between nucleic acids and host proteins or other molecules is also critical to understanding the underlying biology. So for engineered nucleic acids like a DNA or an RNA aptamer, we can quantify the interaction so that we can help produce really sensitive or high affinity biosensors. Or you could just be looking into the basic biology of the interaction to try and understand a structure-function relationship between a DNA and a protein. And so light scattering is going to be a key tool for these types of analyses. We know that traditional methods for characterizing DNA and RNA often revolve around running a gel like this. You watch the migration of your DNA or RNA relative to a ladder, and then maybe you titrate different amounts of a binding ligand or of a drug and watch the change and the shift in the migration. This is a useful tool, but at best it's really semi-quantitative, and it does require labeling your DNA or your RNA with something like a fluorescent tag or a radio label, and that can introduce an artifact in the type of interaction or the type of molecules that you're measuring. Light scattering methods, on the other hand, provide absolute molar mass in solution. So there's no molar mass standard, there's no labeling required, and you can directly measure both the molar mass of your molecules and the interactions and the interaction strength between your oligos and their binding ligands. There are several ways that light scattering techniques can be applied to DNA and RNA characterization. MALS, DLS, electrophoretic light scattering can all be used to characterize physical properties. So this includes size, molecular weight, and charge. These are well-known measurements and they've been applied to many molecular and particle systems. I'm not going to talk about this in too much detail in this webinar, and I'm going to refer you to some of our previous webinars and application notes to discover how light scattering can be applied to nanoparticle characterization. Instead, I want to focus on some unique applications of measuring molar mass of nucleic acids and their complexes. In the first case study, we'll look at molar mass data similar to this structure paper and use it to characterize the complex of a DNA and protein. And then in the second case study, we'll use CG mols to measure the affinity and stoichiometry of the complex in solution directly. So how does this work? The case studies in this webinar are going to revolve around multi-angle light scattering, or MALS. This is also known as static light scattering. For this measurement, we illuminate the scattering volume with a laser. Most of the light passes all the way through, but some of the light is scattered in all directions by the molecules that are in the solution. The intensity of that scattered light is proportional to the molar mass, the concentration, and the refractive index increment, or DNDC. So if you know or can measure the concentration of your molecules, you know their DNDC, and you measure the light scattering intensity, you can directly calculate the molar mass of those molecules in solution. This is a first principles measurement, and it doesn't require any assumptions about the shape or the density of the molecule, and it doesn't require any molar mass standards. We measure the light scattering at multiple angles simultaneously. That's why it's called multi-angle light scattering. And we do that because the variation in the scattering angle is proportional to the size of that molecule. So molecules with a radius less than 10 nanometers will scatter light evenly in all directions. We call this an isotropic scatterer. The DNA and the protein examples that I'm going to show fall into this category. As molecules and particles get larger than 10 nanometers, they scatter light anisotropically. So you get more scattering in the forward direction and less at the back. And you can measure this variation in scattering angle to determine the RMS radius, or the RG, of your molecule or particle. This type of analysis is really critical for understanding larger DNA and mRNA molecules, and also for nanoparticles that might contain DNA or RNA therapies. In my first case study, I'll show you how MALS is incorporated into a standard size-exclusion chromatograph. So here we have a typical configuration with an HPLC or UHPLC and the appropriate columns. The effluent is flowing into a UV detector, our multi-angle light scattering detector, and a refractive index detector. The UV and the RI detectors are what's going to provide the concentration measurement. The MALS intensity then goes with those two concentration measurements to give us a molar mass. So here's a typical chromatogram. We can see the light scattering intensity here in red and our two concentration measurements here, the UV and the RI. At each eluting data point, like this one, we combine the light scattering intensity and the concentration to give a weight average molar mass. This red trace is actually the combination of 18 different light scattering detectors. And so here we see the light scattering intensity as a function of angle. The slope of this line is related to the RMS radius, or here we get 11.5 nanometers. And the intercept is what gives us the molar mass of 122 kilodaltons. What this means is that we're able to measure the molar mass all across a peak without relying on any molar mass standards. It also means you can determine a true distribution of your molecules in solution. So you might have discrete populations like this BSA, where you have a monomer, a dimer, a trimer, and each population is very monodispersed. There's a flat molar mass profile across each peak. Or you might see a continuous distribution like this dextran. This is more typical of things like a polysaccharide. And so you get a continuous change in the molar mass, and you can characterize things like the weight average or the number average molar mass moment. For both of these cases, the molar mass is measured directly and independently of the elution time and the elution order. And moles is really crucial for measuring the molar mass in chromatography mode, even when the separation happens under ideal SEC conditions. So here's an example of two chromatograms. The red trace is a molar mass standard, and I've overlaid the measured hydrodynamic radius on each peak. So this larger one is about 15 nanometers. This smaller one is about 8 nanometers. Our blue trace is our sample. I've overlaid that measured hydrodynamic radius as well. You can see our sample is quite polydisperse, but the separation is ideal, meaning that the part of our sample that elutes here at 26 minutes is the same size, about 15 nanometers as our standard, and the part of our sample that elutes here around 29 minutes is also the same size, or about 8 nanometers. The SEC is working exactly as we'd expect. Now let's look at the measured molar mass that we get from moles. Here's where we run into some issues where the molar mass of our sample does not match the molar mass of the standard. Here, we can see that the 15 nanometer standard has a molar mass around 200 kilodaltons, where our sample is closer to 500 kilodaltons. This mismatch is because the sample and the standard have a different density and a different conformation. So even though the separation is ideal, it's according to the size that we would expect, the molar mass that you might estimate just based on the elution time is significantly lower than the true molar mass that we measure with moles. And you can instantly imagine how this might be applied to a DNA or an RNA sample. These are highly charged molecules that don't have the same conformation or the same density as, say, a protein molar mass standard. And so having a moles detector means that you can directly measure those molar masses without worrying about not having an appropriate standard for your molecule. So all these ideas are well and good for a pure species, like a pure protein or a pure DNA. But what happens if you have a complex, something like a pegylated protein or an RNA conjugated to a polysaccharide dendrimer? So here's an example of a protein whose monomer is expected to be 85 kilodaltons. This solid line is the UV chromatogram, and this dashed line is the DRI measured at the same time. I've left the light scattering off just for clarity. So for a simple molecule, we might get the concentration from the UV detector using our known extinction coefficient, or we might get the concentration from the RI detector with the known DNDC. Then we combine this with the light scattering intensity to get the molar mass. But this doesn't quite work for a complex. So here's the same chromatogram, and now I've overlaid the measured molar mass using our two different types of calculations. So with these blue diamonds, we're using the concentration from the UV detector. We're using our known extinction coefficient and a DNDC that we use for a pure protein of 0.185 milliliters per gram. Our measured molar mass of 110 kilodaltons is not the monomer, and it's not any oligomer. It's clearly not a dimer or trimer or similar. We also see that this molar mass is pretty flat. It's constant across the peak. So we don't think we have monomer co-eluting with dimer. It's a very monodisperse kind of a species. Okay, so maybe our extinction coefficient is wrong. So we tried this same analysis using the RI detector as our concentration source. In this case, we get a molar mass of 98 kilodaltons. Still not our monomer molar mass, still not a oligomer or something that we would expect to happen. So clearly something is wrong, not only with our concentration source, but also with our DNDC, with the understanding of what the composition of this molecule actually is. In this case, this is a glycosylated protein, and we don't know the degree of glycosylation. We need to measure that with SEC mols. Fortunately, we have a solution that allows us to measure the amount of each component in our complex directly. This is called protein conjugate analysis. We have a complex molecule with two components. In this example, it's a protein and a glycan, but it could be two of any polymer. So each component has a different extinction coefficient and a different DNDC. We measure the concentration now using both the UV and the RI detector together. This gives us the mass fraction of the protein, the mass fraction of the glycan, and the molar mass of each component. So there are multiple applications for this technique. And you can imagine in general, these types of conjugated molecules are notorious for not eluting at the right time. So that means having this kind of analysis and having mols is really crucial for determining the right molar mass. So examples are things like glycosylated or pegylated proteins, or the opposite, having a polysaccharide functionalized with peptides or with nucleic acids. The complex doesn't actually have to be covalent. We could measure the amount of lipid or amount of detergent that's associated with a membrane protein. And this is really important for determining the native oligomeric state of that protein, or for choosing a mobile phase that will promote that native state. And of course, we can apply this to complexes of nucleic acids reversibly bound to proteins. If we go back to our previous example, we can see that applying protein conjugate analysis gives us the right answer. When we use both concentration detectors, we can separate the protein part from the glycan part. Here we see the protein molar mass is 85.8 kilodaltons, which is exactly what we expect for the monomer. And then we have an additional 15.6 kilodaltons of glycan, making the total molar mass 101 kilodaltons. Now let's look at how to apply this SEC mols and protein conjugate analysis to a protein DNA complex. This example looks at the prototype foamy-virus integrase, or PFV integrase, in complex with some viral DNA. The original data, along with complete structural information, was previously published by this group at the University of Pennsylvania. The first step in doing this analysis is to confirm the properties of the individual components. So here we have a chromatogram for the PFV integrase, along with a cytochrome C control. The UV trace is the solid line, and the RI trace is the dashed line. And again, I've left the light scattering off just for clarity. By measuring both the UV and the RI simultaneously, you get a fast and direct measurement of the extinction coefficient for that molecule. I want to point out that you can do this at any wavelength. For this protein conjugate analysis, we used the extinction coefficient at 280 nanometers for both the protein and the DNA. But you could easily measure the extinction coefficient of this protein at 260 nanometers and use the same 260 nanometers to measure the DNA part as well. Okay, so first let's look at this cytochrome C control. The predicted extinction coefficient based on the sequence is 1.12, and using our simultaneous UV RI measurement, we get a measured extinction coefficient of 1.08, which is a difference less than 4%, and that means that the sequence value is a valid measurement. When we do the same thing for the PFV integrase, we get an extinction coefficient from the sequence of 1.34, but we actually measure an extinction coefficient closer to 0.75, which is almost half of what we would expect just based on the sequence. That's something we wouldn't have known a priori, and which would have significantly impacted our ability to measure the molar mass accurately. Now we confirm that this extinction coefficient is correct by measuring the molar mass, both with the Ri concentration and the UV concentration. And in this case, both of these give us the same expected molar mass of 44 kilodaltons for that monomer. Now let's look at the complex of the PFV integrase bound to the U5 DNA. The complex is called the intosome, and it elutes as a single peak. We are showing again the UV and the Ri trace. I've left off the light scattering for clarity. So we're going to apply the protein conjugate analysis to this whole complex and see how much protein and how much DNA we actually have. First, we'll input what we know about each species. So that's the measured extinction coefficient of the protein and the DNA, and also the refractive index increment, or DNDC, of each part. Our Astra software then uses the light scattering, the UV, and the Ri to calculate the molar mass and the mass fractions of each part at every point across this chromatogram. So here is the light scattering in red, the UV in green, and the Ri in blue. At this instant, here around 27 minutes, we calculate that the protein fraction is about 90%, meaning 90% of the mass is protein and about 10% is DNA. Based on this information, we can get the total concentration at that slice. We can get the DNDC for the entire complex and the extinction coefficient for that entire complex. We use this concentration in this DNDC now to measure the molar mass. So here is that light scattering intensity as a function of scattering angle. The intercept is giving us the total molar mass at 194 kilodaltons. The protein part makes up 174 kilodaltons, and the DNA part makes up about 20 kilodaltons. Since we're calculating all this information about the conjugate at each slice, we can overlay the protein, DNA, and total molar mass all the way across the peak. This characterization that we're doing in solution can now help confirm the complexes that are seen in the crystal structure and other orthogonal measurements. So here we see the protein molar mass reaches about 170 kilodaltons, which is a tetramer of this integrase. That tells us there's 4 integrase proteins coming together in this complex. The DNA molar mass reaches up to 22 kilodaltons, which tells us we have 2 double-stranded oligos per complex. This combination of 4 proteins and 2 DNA strands is exactly what we see in the crystal structure. So this confirms that the structure is relevant in solution and not just a crystallization artifact. You might also notice that the molar mass is not constant across the peak. There's a slight downturn. So that might mean that we have on-column dissociation. So the maximum concentration at the apex of this peak is only about 7 micrograms per milliliter, which is less than 40 nanomolar of complex. So depending on the KD of this interaction, we might be seeing some species that are not saturated, co-eluting with some fully bound species. In our next case study, we'll explore more about how to measure these kinds of affinities and stoichiometry in solution. In this case study, I'm going to present a different application of mols called CG mols, or composition gradient multi-angle light scattering. This lets us measure the affinity and the stoichiometry of complexes forming in solution. In this case, we're looking at a Cre recombinase in complex with a LOXP DNA recognition site. We've already seen how to apply mols to a fractionation system like SEC. CG mols is applied to batch or unfractionated samples. So to do this, we create a series of different compositions and inject them into our light scattering and concentration detectors. The mols detector then determines the molar mass at each composition, and we can relate the change in the molar mass to the formation of complexes in solution. We automate this type of measurement with our Calypso instrument. Using this technique, we can assess self-association, so like monomer-dimer, trimer, equilibrium. We load a single stock concentration onto the Calypso, and then it takes an aliquot of the sample, an aliquot of buffer, mixes it together, sends it into our detector, and stops the flow. We measure the molar mass as a function of different concentrations and use that to determine the self-association, affinity, and stoichiometry. So if nothing happens, if no complexes form, the molar mass will stay constant as a function of concentration. As complexes like dimers and trimers form, we see an increase in the molar mass as a function of concentration. We can fit this increase to an appropriate model to tell us the affinity and the stoichiometry that we're seeing. We can do the same thing for an interaction between two different species. In this case, the Calypso will take an aliquot of each one, mix it together, send it into the detector, and stop the flow. This type of experiment design is different from a titration. Each of these plateaus represents a fresh mixture of A and B that haven't seen each other before. We're also not going to keep the concentration of either of them fixed. Instead, we'll have regions where A is in excess and regions where B is in excess, and that lets us sample the entire stoichiometry of that interaction. The result is a characteristic curve, something like this. So here we have the light scattering intensity. On this left axis is the signal that we would see from pure species B. As we walk from left to right, we're decreasing the concentration of B and simultaneously increasing the concentration of A. If nothing happens, if these molecules don't bind, the light scattering intensity is just the weight average of the two species. As complexes form, the molar mass of that solution increases, and we see a characteristic curve like this. The more complexes bind, the stronger the interaction, the higher and sharper this peak becomes. The position of this peak is related to the stoichiometry of the solution. So if each B has one binding site for A, we'll see an apex at the 1-to-1 molar ratio. If each B can bind two A molecules, we'll see the maximum at the 2-to-1 molar ratio, and so on. The advantage of this CG-mols technique is that we're measuring molar mass directly. So we can tell the difference, say, between a 1-to-1 and a 2-to-2 interaction. They'll both reach a maximum at the same position, but the 2-to-2 has twice the molar mass, and we'll measure that directly. To study the interaction between Cre and LOX-P, we do a three-part experiment. In the first part, we have a concentration gradient in the Cre. This lets us measure the molar mass of that protein alone and determine if it self-associates. The last part of this experiment is a similar concentration gradient for the DNA by itself. So we'll confirm that molar mass and see if we have any self-association of the DNA by itself. For each molecule, we're measuring the light scattering intensity at 5 different concentrations, up to a maximum of about 20 micrograms per milliliter. We can immediately see from these data that neither molecule is reversibly self-associating. So our Cre molar mass is measuring a constant 39 kilodaltons, which is consistent with the monomer. And our DNA molar mass is measuring a constant 23 kilodaltons, which is also the monomer of that oligo. If either of these formed some kind of reversible association, we would expect to see the molar mass increase across this concentration range. The fact that it's staying relatively constant means that they're not self-associating at all. The center part of this experiment is our heteroassociation gradient. In this case, we create 13 different mixtures of the protein and the DNA to quantify the interaction between the two molecules. For each of these mixtures, The calypso is going to take a fresh aliquot of the protein, a fresh aliquot of the DNA, mix them together, send it into the detector, and stop the flow. We allow this mixture to come to equilibrium inside of our moles flow cell and use the equilibrium molar mass for the analysis. I want to point out that for all 23 data points across this whole experiment, we're only using about 200 micrograms of each species. So here's what the results look like. In this graph, we see the light scattering intensity in blue overlaid with the concentration of each species. This solid black line is the concentration of the Cre protein, and this dashed line is the concentration of the LOXP of the DNA. In the middle part where we have our heteroassociation gradient, we can immediately see that we have some slow kinetics going on. That when we mix the two of them together, it takes some time before the molar mass, before the light scattering intensity reaches a final equilibrium. Here in this inset, we're going to look more closely at these data points where we have about 10 micrograms per milliliter of each species. Here I've highlighted where the solutions are flowing into the detector for the first 30 seconds or so. After that, the flow is stopped and the solution is allowed to come to equilibrium. So this increase in the light scattering intensity is proportional to the increase in the molar mass over time as the Cre-LOXP complexes are being formed. And this comes to equilibrium over the course of about 15 minutes. So now we'll take a look at the equilibrium molar mass for each of the plateaus that we created in the previous slide. Each data point on this graph represents the weight average molar mass at equilibrium for those different compositions. On this left axis, we have the molar mass of the pure Cre at around 40 kilodaltons. As we move from left to right, we're decreasing the concentration of the protein and simultaneously increasing the concentration of the DNA. This molar mass on the right-hand axis of 23 kilodaltons represents the molar mass of the pure DNA. This maximum of around 110 kilodaltons immediately tells us that our stoichiometry is greater than 1-to-1, right? 40 and 23 are much less than 110. So now let's look at the binding in more detail. This red line shows the predicted curve, the predicted molar mass, if only one protein binds to each DNA. The maximum molar mass for this model occurs at a 1-to-1 molar ratio where we have equal amounts of Cre and LOXP DNA. Obviously, the maximum possible molar mass, around 60 kilodaltons, is much less than what we actually measured. Also, the position of the maximum is in the wrong location. So we need a model that has a higher ratio of Cre to LOXP. So what about a model where we have two Cre proteins bound to each LOXP DNA? This green curve assumes that each DNA contains two binding sites and that all of the available binding sites are always filled. So this is a completely saturated curve. This model has the apex in the right position. So at the 2-to-1 molar ratio is where our data also reaches a maximum. However, this still doesn't fit. There's still some missing element in this mechanism. So first, even assuming that all of these sites are saturated, we're still underestimating the actual molar mass that we measured. So this suggests that there's higher order structures being formed. It's not just our usual 1-to-1 and 2-to-1 complex. Second, we can see some curvature at the high ratios of DNA to protein that is not being captured by a simple 2-to-1 model. So fortunately, since we're measuring this molar mass directly, we don't actually need to make any assumptions about the binding sites in order to fit this data. We can let the data be our guide to the right binding mechanism. So the right fit requires 3 different complexes. The first one is one Cre protein bound to the DNA, and this first binding occurs with an equilibrium dissociation constant of 170 nanomolar. Then a second Cre binds to this complex, and we find that this second event is cooperative. So having one Cre protein bound to the DNA increases the affinity for the second one, and we see a 10-fold reduction in Kd, and the second binding event happens with a Kd of 17 nanomolar. Finally, this Cre-LOX-P heterotetramer dimerizes in a process called synapsis. Under these conditions, we find that the dimer forms with an affinity of 400 nanomolar. So now with a single CG-mols experiment exploring concentrations of just a few hundred nanomolar for each species, we're able to describe this 3-step binding mechanism fully. We don't have to work in multiple concentration regimes or assume that one or more of these binding sites is fully saturated. Most importantly, I think, is you don't need to assume that one part of this reaction is negligible. You can get complete characterization and measure all of it with just a single experiment. Not only do you get the affinity and the stoichiometry, but CG-mols also gives you the composition of the solution for each mixture. In this graph, I'm showing the fraction of each complex that's being formed. I've left off the fraction of the free monomer for clarity. We can see, for example, that at a mixture of about 475 nanomolar Cre and about 200 nanomolar LOX-P, we get a solution that is 35% of this 2-to-1 complex, about 10% of the 1-to-1 complex, and about 10% of this synapse tetramer, and the rest is free monomer. This type of an analysis can really help with interpreting orthogonal measurements like cryo-EM or other structural analyses. And we have a few examples of these types of measurements in our bibliography, and I encourage you to look at those. So to sum up, I've hoped I've shown you that light scattering, either with fractionation or in batch mode, is not just for proteins. These same analyses are critical for DNA and RNA and for characterizing nucleic acids in complex with other molecules. In this webinar, we explored 2 different kinds of characterizations for a similar protein DNA system. SEC-mols with protein conjugate analysis gave us the absolute molar mass and the protein and DNA fraction for a viral integrase bound to its DNA ligand. This is done without molar mass standards and without assuming anything about the stoichiometry. On the other hand, CG-mols in batch mode without chromatography let us quantify the affinity and the stoichiometry of that complex in a multi-step protein-DNA interaction. You can find more information about these stories and other applications on our website, and perhaps they'll inspire you to think about incorporating these techniques into your own characterization. With that, I'd like to thank you for your attention, and I'm happy to answer questions.
So here's the first one. Can we determine molecular weight or molar mass without knowing the sample concentration? Sophia, are you there?
So sorry about that. So the light scattering intensity is proportional to the molar mass times the concentration. So in SEC mols mode, we typically have an inline concentration detector, either a UV or RI or both, that's giving us that concentration information. For CG mols in batch mode, you can have an inline concentration detector, or you can determine the concentration of your samples offline separately and then use the calculated dilutions for the analysis.
Okay, thank you. Here's the next question. How do you measure extinction coefficient for single components in Astra?
So it's actually really easy in Astra. When you are doing SEC mols and you have both a UV and an RI detector, chances are you do know the DNDC of your single species. And so there is a method in Astra called UV extinction coefficient from RI peak. And so it takes the measured refractive index and the measured UV signal and matches the two of them. So if you know the mass from the RI, it can back calculate the extinction coefficient in the UV detector.
Okay, thanks. Next question is, how accurate is a KD value obtained from CG mols?
So we actually get really good agreement for the KD with CG mols as with other techniques like SPR or ITC. The nice thing is that the CG mols is done in solution. So where an SPR technique or something similar where you've got one of your analytes bound to the surface can have some surface effects or some avidity effects. We don't have that issue. So ours is usually very consistent with the solution-based techniques like calorimetry or centrifugation. And we'll usually get reproducibility somewhere between 10 to 30%.
Okay, thanks. Here's your next question. So how do differences in DNDC for proteins versus nucleic acids influence analysis?
So the molar mass that we measure from light scattering is proportional to the DNDC squared. So by knowing that value, either for proteins or nucleic acids in the solvent of interest, so in your buffer solution, is how we are able to measure the molar mass. And the fact that they are different for the protein and the nucleic acid lets us do that protein conjugate analysis that gives the amount of each species coming off of a peak. And so what the value is doesn't influence the how the measurement is done or the accuracy of the measurement, but knowing it is required for getting that molar mass.
Okay, thank you. So is there a generic DNDC value for DNA, RNA, much like there is for a protein? And how accurate do these values need to be for conjugate analysis?
Sure. So for the examples that were in the webinar, we used the literature value of 0.17 as the DNDC for nucleic acids in water. And we've had really good luck using that in other applications as well, not just the ones that were in the webinar. The error in the molar mass is going to be proportional to the error in the DNDC, but typically for aqueous buffers that don't have a lot of extra excipients or other things in them, you can use that 0.17 value for nucleic acids and the 0.185 value that we typically use for proteins with good accuracy.
Okay, thanks. Next question is, how can we measure the hydrodynamic radius of this DNA protein aggregation?
Sure. So we use dynamic light scattering to measure the hydrodynamic radius typically. So within the Helios or Trios, within the light scattering detector, we can embed a dynamic light scattering detector. So we're simultaneously measuring the hydrodynamic radius with dynamic light scattering, as well as the molar mass with static light scattering. And so that can give you a size in addition to the molar mass. And we could do that for both the chromatography mode with the protein conjugate analysis and the batch mode with the CG mols. We could also use an inline viscometer, so like our ViscoStar, for hydrodynamic volume in online mode with SEC. So either one of those can give you size.
Okay, thanks. So how do the equilibrium constants determined by CG mols compare with those obtained by surface plasmon resonance, isothermal titration calorimetry, equilibrium sedimentation, or other techniques?
So in general, we get really good agreement between the constants that we get from CG mols and other solution-based techniques like analytical ultracentrifugation or ITC. So we've had studies and we have customers that use a variety of techniques and can compare the two of them together. We typically get also very good agreement with SPR as long as, like I mentioned before, as long as the surface isn't influencing that interaction. So as long as your analyte isn't either non-specifically binding or caught in some kind of avidity effect.
All right. So if I'm analyzing a protein DNA complex by SEC, should I use protein calibration standards or DNA calibration standards?
So with mols, you don't have to use any calibration standards. You're going to be measuring that molar mass directly, and so you don't need to worry about how it runs on the column relative to either a protein or a DNA standard. So we'll just get that molecular weight, and you'll also be able to see the composition using that protein conjugate analysis.
All right. For next question, Sophia, I'm going to point you to our Q&A thing. It's a little bit complex. So would the CRE IOXP 4:2 stoichiometry be more observable with longer binding times in the CG mols experiment?
When we were performing this experiment, we did see that the equilibration time was rather long. So we took, I think, either 15 or 20 minutes to allow that solution to come to equilibrium. And over that time, what we saw in the light scattering signal is that at early times, the signal would increase, and then eventually it would saturate, and that it saturates as we approach equilibrium. And at that point, you could keep waiting, but the concentration of the products and reactants is where it's going to stay for that equilibrium. So watching it longer wouldn't cause more of those species to appear. What would cause more of those species to appear would be to maybe change the concentration of either the CRE or the LOXP and have more material available for binding, and then we might see more of that 4:2 stoichiometry present.
Okay, thank you. So how does the analysis depend on molecular conformation? Can it determine the geometry of a protein DNA or DNA complex? So, for example, can it discriminate compact from elongated geometries?
So all of the examples that we had in the webinar were isotropic scatterers, which means that we, with mols alone, are not able to measure a size. In general, the mols measurement, getting the molar mass and getting the conjugate analysis or getting the binding site is independent of the conformation. So we would get the same result whether it's compact or elongated. Adding a size measurement like a DLS or an online viscometry would give us additional information about the hydrodynamic size, and so we could draw some conclusions about the conformation, if it is compact and globular or if it's more elongated. But the conformation doesn't impact how we do the analysis.
All right. Well, thank you very much, Sophia, for your presentation and for taking the audience's questions. I'd also like to thank the audience for attending and participating in today's event. And I'd also like to thank our sponsor, Wyatt Technology, for making today's webcast possible.